So with that, I'd like to introduce our next speaker, Jonna Mazet, a professor of epidemiology and disease ecology at the UC Davis School of Veterinary Medicine. And Jonna actually was at the last meeting, I think, that we hosted with CEND, the in-person in January, early January 2020. And this was the very first time I heard about COVID, because Jonna and some other people in this field, Heinz Feldman, also were chatting during coffee break about this new agent that got just reported from China, and they were very worried about it. And honestly, I hadn't really heard that much about it before, and there was still a sense maybe this is not going to be so bad. But I thought back often in the last year to this coffee break interaction, because the experts were clearly very worried about it, and that taught me something about how we need to take this seriously. And also that Jonna has been part of this group of people who have warned about this for a long time. In fact, she has been the global director of the PREDICT project for more than 10 years, early viral emergence warning project. She's passionate about the One Health approach, which integrates human health with veterinary health and also environmental factors, understanding that we're all part of the same environment and viruses can jump from animals to humans, and focuses now with the Global Virome Project on making sure that we have better tools for pandemic preparedness and better responses. So welcome, Jonna. Great to see you again.
Thank you, Julia. It's great to see you. And I might have some of the same gremlins you had earlier, so we'll see how we do with the screen wants to change on its own. But yes, I was reflecting this morning about the last time I participated in a CEND Symposium on January 10th of last year, obviously 2020. And we actually, the panel included a person from Gilead. We were talking about remdesivir and coronavirus vaccines made for cats. So yes, who knew that we would be in this situation all of this time later. So thank you for having me. I have spent most of my, at least more than the last decade of my career on early warning systems for pandemic preparedness and finding viruses ahead of pandemics. So it's bittersweet to be with you here after sheltering for more than a year and finding ourselves in this terrible situation. But I do agree with the colleagues who have gone before that we have the ability to get out of this one and to never be in this situation again. And I hope to build on their great comments and share with you some of my thoughts about how to do that. Certainly, I'm here to talk to you about using the One Health approach, and it's much easier to do that now than maybe it was last time we met, in that the One Health approach is something that is talked about in the mainstream media these days, of really thinking about humans, animals, the environment, plants, how we all interact and how our health is shared. That said, that did not help us to find COVID-19 causative agent, SARS-CoV-2, ahead of this one. We were not working in the part of China where the pandemic really started to take hold, where it was first an outbreak in humans and then it became a regional outbreak and then a pandemic. But we did identify many of the risk factors and coronaviruses specifically as an issue. And I will take you through that. Why I'm talking about One Health and the interface of humans, animals, and the environment, especially for zoonotic diseases, is because that the majority of emerging infectious diseases in people are of animal origin, and 3/4 of those are continuing to evolve in wildlife hosts. Now, these viruses don't leap out and grab us, and we also aren't attacked by bats. Very rarely. We could talk about that if you want, but it's very rare. This is really a consequence of our own behavior, our own human footprint on the planet. And we have the ability to think about and change our behavior to reduce our risk from emerging infectious diseases. So just since Julia did ask me to come to you from a One Health perspective, I wanted to throw the current working definition for One Health that I think most all of us are very happy with, and finally unified around this collaborative, multisectoral, and transdisciplinary, meaning we can do more by bringing the disciplines together than we can separately, and it's more than additive, it's actually synergistic and goes beyond. And that's working at the local, regional, national, and global levels. And we've heard about failures of doing that today. And I hope that we can talk about how to move forward to positively affect change that does go from local to national to global. And the goal, of course, is achieving optimal health outcomes, recognizing this interconnected, integrated health that we are all living. So we are vulnerable. I mentioned that there are many diseases that are impacted, and as diseases emerge at this One Health interface, we are seeing more and more. When I started in this realm, the estimates were 3 new emerging infectious diseases of people each year. We're up to 5 now every single year. Obviously, most of them do not take off and cause horrible pandemics like COVID-19 from SARS-CoV-2, but we do need to be ready for and change our thinking and our paradigm to not just be chasing whatever happened to us before. I truly believe that if we allow ourselves, allow our community here and globally to fall back into the same kind of headspace that we have been in since the beginning of time, where we worry about what's right in front of us and what just got us, we will miss what's coming next. And we have the ability to do much, much better than that. You heard about the scientific abilities. You heard about the necessary political will. I completely agree with those. But I also think that we need to integrate our systems and be prepared for disease X, not just the list that we know is coming or has come before. So that said, we can't eliminate every single spillover or every disease outbreak, but we can be ready to identify them and contain them at their source and hopefully mitigate so that we don't get large-scale epidemics and pandemics. Now, when I started this work, there were about 240, now 260 ICTV recognized zoonotic viruses able to make people sick. We have estimated, based on our work and the work of others in the large body of literature, that there are probably on the order of around 500,000 viruses yet to be discovered. And if we can discover them and see what is similar about them, also see which ones are infecting people and what are the differences between those that are making people sick and those that aren't, we really have the ability to change the whole paradigm of being sort of just sitting and waiting for the next pandemic. Now, I do advocate for, I want us to be ready for that next pandemic. We need to have all the systems in place, but at the same time, we can reduce our risk by understanding what's out there and how we interact with those hosts and those viruses to reduce our risk. This is getting to be kind of tired for me. I hope not for you, but it is a good descriptor. Ignore days at the bottom. This is a classic epidemic curve, the red being the human epidemic curve, but we know now that things are not generally diagnosed on the order of days. And depending where you are and the resources available, the public health systems, the laboratory systems, this might be not only weeks, it might be months that these timelines happen over. And what we want to see is we want to understand what's happening in the green area, what we call the zoonotic pool, where these viruses are living happily in their host, generally not making their hosts ill, spilling over into other hosts, could be other wild animals, and we see some spikes of illness, but more frequently that does not get detected. And it's more when it spills into livestock or into humans directly that we see a problem. Now with MERS, that's a great example that was spoken about today, that we can see a spillover happen quite a long time before it is documented to be spilling over into humans. And another thing that I'd like to share with you today is that spillovers are happening very, very regularly, and we just don't diagnose them. Most fevers go undiagnosed in people. That's here at home, as well as everybody else's home that's speaking to us today and living around the globe. And so those spillovers those fevers can be actually often likely spillovers, because we're detecting serologically with new serologic techniques that allow us to do pathogen specific work, that there is a very high level from my headspace of spillovers. For example, in some of the communities in Uganda and DRC that have a lot of hosts with Ebola viruses, we're detecting up to 10% prevalence in people that did not know that they ever were exposed or were made sick to Ebola. So this starts to tell us that what we are really have been detecting are these high peaks, that we have been undetecting low-level disease. Sometimes people die, but their whole family doesn't get sick and die, so we don't detect them. And sometimes they just get better. So what we really want to see is better forecasting. And I've been working for, as Julia said, more than a decade to start to improve the data to do better forecasting, but it is still just the very, very tip of a huge iceberg. And we need the world to work together to do that, to find these viruses and understand not just their genetic sequence, but their whole epidemiologic and ecological characteristics and how people might interact with them and become sick. Then we need the laboratory systems to be ready for early detection, so that anywhere in the world, people can pick up, diagnose, detect viruses that are known to make people sick and viruses in patients or animals that are potentially making them sick but have never been diagnosed before. And that's what will really improve our ability to respond. And I come actually from early in my career, I was an oil spill responder. And so I come from this emergency response training and background. And we should always think about that in health events as well, that we should over-respond early, right? That was a huge problem here. We under-responded. We thought it wouldn't come. We talked about it, in Berkeley, and yet we all felt a little bit comfortable. Oh, that's bad. We should be worried. But our systems did not get excited. We knew actually the virus was in the Bay Area then, right? We know now historically that we've gone back to those cases and the virus was already with us. We need to be thinking and responding early. And I do agree with past President Napolitano that we can and should treat this in a way just as any kind of security threat. So I mentioned I've been proud to be the leader of a huge consortium, hundreds of people in 35 countries. I've listed some of the global partners that help to lead as well, from the modeling, from the intervention perspective, but it was really the university, NGO, and ministry partners in the 35 countries where we work that implemented this amazing project that provided the proof of concept that we can get in front of these problems and respond also well from a One Health perspective. These are the responses that we did as a PREDICT team at the request of the governments. Again, we worked primarily in Southeast Asia and Africa with early years in Latin America. But the majority of the outbreaks that we were asked to help with were really because there was apparently a human-animal interaction, or it was a disease of undiagnosed high consequence, causing hemorrhagic fever, causing severe respiratory illness. And our teams jumped in and not only did human contact tracing, but also environmental sampling, also animal contact surveying. And I think we really helped to strengthen the whole outbreak, PP, everything response in the countries where we worked, learning together as we went. These are the kinds of places, though, where we did that work that was getting us to forecasting. I sometimes call this the flu of death kind of slide, because for a lot of audiences before this year, I have to be honest with you, nobody wanted to hear much about anything but influenza. Right? And influenza is critically important. Of course, we cannot now just switch to SARS-CoV-2 and forget about influenza, but lurking here in this same interface, the bats and the rodents and the shrews that you can't see, and that this is right outside the curtain doorway of the household that allowed us to sample not only their animals and their soil here, but also went into the back of their homes and sampled themselves for us so that we could start to triangulate. This is the kind of detailed, non-glamorous work we really need to do, because the amazing people that do the genetic analyses and all of that hard work that help us to do forecasting have no data to base that on if we don't get out here and find the viruses early now. I guess that's what I want to encourage us all to do. This is how we started. We started with probabilistic models, again, not with the best data available. Well, it was the best data available, but unfortunately, we know we need so much more. That helped us identify the highest priority sites to do field studies, and in which communities we could empower and engage and partner with those communities. And I think community engagement is also critical, and something that in science we've done pretty poorly for the past couple hundred years. And I'm seeing a lot of improvement now because those communities are ready and able to be raising the flag early, and we should be listening and ready to respond. Then getting those samples into the laboratory, for us, that was difficult because, as we've heard earlier, many of the very expensive systems weren't available where we're working. And even if they were, they aren't necessarily proving to be the absolute best from a perspective of sensitivity for really picking up novel virus that may be at low copy number and other things. So we had to come up with a strategy that would work in any resource level setting, and then get that data back into the models to start improving them. And I'm pleased to say more than a decade later, we have great evidence that that is working. These are some more interfaces where we worked. And I think to speak to Dr. Varmus's point, the urban center that you see on the right, we are living much more closely with animals and wildlife than we often acknowledge, and that we don't acknowledge the risk. And when those viruses either spill over in an urban environment or they move into an urban environment in the wildlife trade or in a migrating animal or in a person who is asymptomatically carrying or even coming to go to a better urban center to go to see a physician, then we have the ability for it really to take fire. On the left there is just to remind me to talk to you about new burgeoning economies and livelihoods. This is bat guano farming. And what we know from working with bat guano farmers is that many put these bat attractants that you see here, bat condos, I sometimes call them, right above their crops, and allow the guano to just go onto the crops, and they go straight to market with those crops. Others are thinking about, how to disinfect. So you see the UV there in the lower corner, basically laying it out to help disinfect. Does that work? We can't just tell people, stop doing that scary livelihood practice. Please don't use bat guano. We know it has coronavirus in it. We have to say, what's in it? Work with the community, look at different practices, see if they're being safe for themselves, because it doesn't necessarily have to be on the crop that's the problem. If the bat guano harvester themselves get sick, then they could start to spread, right, and start an epidemic or a pandemic. And of course, I'm going to talk to you about bats today. We spent a lot of time sampling bats, non-human primates, and rodents. We had lots of other samplings as well that those samples ended up going into the freezer to be pulled out during COVID, like from pangolins and other things, to try and find the evolutionary host of SARS-CoV-2. And of course, when we think about bats, some of you probably don't want to think about bats at all, but they are incredibly important parts of our ecosystem. This is a tree in an urban area. And even when we go to urban areas, often people talk about not really having bats when they have millions of bats in their community. And this is often in other places where people like to eat bats and use them for medicinal purposes, that you can pick them up quite readily in the marketplace. And these are traded and moved. And these bats aren't cooked or smoked. When they are, that we found that there is much less viral risk. But these are not. They're just blowtorches used to remove the hair so that they are more palatable as a market in the market to sell. Okay, so what did we do specifically, and how is that helpful for thinking about what can be done in the future, or our sort of charting our path out of this pandemic era? And we were using that pandemic era prior to this horrible pandemic, and that's pretty devastating to me as well, to think that as scientists, we weren't able to convince our policymakers and our communities that this was a real risk. And many famous scientists were agreeing, but it wasn't the top priority for really advocacy. And I think that it's time, obviously, to capitalize on this horrible tragedy and remind people that we can do this differently. Now, what we did was we looked in the viral pool. So I showed you the green area on the previous slide for that early recognition of potentially zoonotic viruses in wildlife. But we also did syndromic surveillance and occupational surveillance. So for example, at that market with the bats, the market stall managers, as well as the people who were harvesting guano or harvesting the bats from the environment. We were looking at healthy individuals to see if we could pick up the same viruses that we saw in the wild individuals, as well as in the hospitals when people were coming in with fevers of unknown origin from those same sampling basins. We were able to start to triangulate. We also worked with WHO on the human side and FAO on the livestock side to start to look at that. Now, of course, when we were looking at the humans and the livestock, we found a lot of known human and livestock viruses, and that's because they've been studied, right? And we've seen them before. We've evolved with our domestic animals, our food animals, and so we know a lot more about those viruses that have been seen before in humans and livestock. So that's not surprising, but it's the ones that that might be being shared that we're more concerned about. To do this, we had to train a biosafe disease surveillance workforce. And so obviously these team members during the COVID response became some of the most sought after experts in PPE and working in the field. They honestly, for many countries, were immediately grabbed and put on their national task force for COVID. And I'm really excited about that for them and for their countries, that they had people that were used to working in the field and the laboratory. We also had to develop non-invasive techniques. And so we worked with primate centers, our primate center in Davis, as well as zoos, to really find the best sampling techniques so that we don't always have to anesthetize and intervene with wildlife to find their viruses. So we came up with techniques that if we anesthetize and swab that monkey's mouth, we would get the exact same thing on that attractant and rope and could pull it back. But we had to get those samples into the laboratory and train a pathogen detection workforce. This is one of the things that I think was most useful in the COVID response, is that there were more than 60 laboratories that were used to doing novel virus detection and characterization. And we used consensus-based PCR to go sort of a low resource method that also turns out to be a bit more like primary virology than receiving a kit. So these labs actually jumped into action before there was even a full sequence known for SARS-CoV-2, and they were able to detect, sometimes in places like Nepal and Thailand, the very first introduced cases in sick people coming in through airports and their border into their countries using the PREDICT protocols. And then, of course, moving to the real-time specific SARS-CoV-2 detection when the sequence was available. So this project just tell you that we were able to train almost 7,000 people in this approach to be biosecure, biosafe, many of them having publications in things like coronaviruses and novel disease detection in their countries. More than 60 laboratories were ready and able to jump into action. We did do a lot of work. We sampled more than 164,000 individuals to find about 1,200 viruses. Now, that's not scratching the surface of the 500,000 that need to be found, but we were flying the plane as we were building it. So we really were able to develop the foundational materials to allow countries. Now we've translated the many protocols. We have a whole book of protocols that are available on our website. It's www.predict.global. You can find the legacy book that talks about what we did and how we did it, as well as the protocols and guides. And you can link to our publication list that probably we haven't updated quite well during COVID, but there's a good start there. And it points to the ability now and having everything we need to find that 500,000 viruses that are out there. Excuse me. Okay, not COVID, fully vaccinated, proud to say. So what did we find? So we found a lot of novel viruses, a lot of novel coronaviruses, but we also started to really recognize that you could identify hosts. You didn't need to find the virus in every single corner that it lurks to inform communities. You could start to find the hosts that were evolutionarily linked with some of these viruses. This one in particular was a MERS-like coronavirus that we found in Uganda. We found it in this bat, and we could map the hosts of this bat, and other closely related bats that tend to roost together with this bat throughout Africa, and look at the sort of connection with the camel trade and how camels get into the Middle East because they're bred quite often in East Africa. And so we started to really understand how we might get at risk mitigation without necessarily having to sample in every interface in every country for every species. Now, I do think that that's feasible and we can do it, but it probably we can achieve our goals much more quickly than trying to dive into every single little spot by starting to understand the virus host dynamics and then mapping those hosts that are much better known than the viruses. We also detected a novel Ebola virus in Sierra Leone. And that novel Ebola virus was found in a couple of species of bats. So here's my example here. The map of Africa there shows you the distributions of those hosts throughout Africa that we found positive in Sierra Leone for this novel, the Bombali Ebola virus. Now, interestingly, right after we put that out into the public domain and worked with the communities and worked with the government, the virus was found in Kenya. So you can see that it's showing you that wherever this map looks to be highlighted, this pathogen potentially, we know that it can infect human cells, but we don't know if it makes people sick, but it should be on the differential list for this entire mapped zone. We also found, while doing that same activity, Marburg in these bats in Sierra Leone. And we worked in conjunction with other sites that were being sampled by the CDC and Njala University. They found it at the same time we found it. So what did we do? We published that together, trying to bring scientists together and really group science a lot of this work to have better outcomes. Now you can see where the previous detected outbreaks of Marburg were primarily in Central and East Africa, a very deadly pathogen, not known to exist here. Now, did it ever make anybody sick in this far west in Africa? Perhaps, perhaps not, but now it is on the differential list. And we know the host, CDC's done wonderful work identifying rosetus bats as the host. And if you map the rosetus bats, look where you should find this pathogen. And that is where we found it. Okay. So this is the kind of work that we can do efficiently if we all start working together. Now, let's talk about coronaviruses. So in working on coronaviruses, working with teams that had worked on the original SARS, and continuing that work in our PREDICT project, looking for hosts of SARS-related coronaviruses and other coronaviruses, we were able to do some really important work on the ACE2 receptor that started to move things in a better prepared direction and were really useful during this pandemic. And we detected 177 coronaviruses. Now, there were only a handful of coronaviruses known before we and others started working post-SARS in this realm. 160 of these 177 viruses that we detected were novel coronaviruses, not yet known to science. And you can see the majority of those were detected in bats. Now, what that allowed us to do was start to look again at the host relationships, at the ACE2 receptor, really building the information base. And what we found were the Sarbeco coronaviruses, the SARS-related coronaviruses, or the 2B coronaviruses were by and large, hugely most of the time found in rhinophilid and hippocytarid bats. Again, that allowed us to go to WHO and other partners and say, let's look at the distribution of rhinophilid and hippocytarid bats to see if we might have ongoing spillover events of SARS-CoV-2. And if you do that, you see that it is up in central all the way to southern China, but almost all of Southeast Asia, as well as all the way over through Nepal, where we should be thinking about ongoing monitoring for SARS-related coronavirus spillovers and what might come. In addition, we were working, as I mentioned, in that human food value chain. I showed you some market pictures, but we could work in this case in Vietnam with trappers and traders who are capturing rodents out in the field and taking them in to farm them or taking them through the human value chain into markets. And we found coronaviruses in about 20% of those rodents out in the field. Of course, as you start to concentrate them, put them in stressful situations, mix them with other animals in that value chain, we started to see that go up about 32% in the large markets. By the time we got to restaurants where people were butchering and eating these rodents, the prevalence was over 50%, showing us how frequent these risks are for spillover of coronaviruses. Really how lucky we are that we haven't seen this again and again. And we need to understand why that is and what makes the perfect storm for a virus to actually get out of control. Now, our teams had stood down. The project was slated to end. We were funded in 2 5-year cycles, and we were finished in the countries when SARS-CoV-2 spilled over, likely, but at least when we detected the COVID-19 outbreak. So our teams were no longer working or being paid by PREDICT, but some, like we heard about just in the talk before me, the Uganda Viral Research Institute, other partners, those ministries were ready and able to jump in. And thankfully, USAID did come back with a funded extension to allow us to help in some of the countries moving forward through the last year. So we did extend at a small level for testing, for looking in the freezers for evolutionary hosts of this virus, and for simply just helping with the infrastructure, travel and trade restrictions, all of those things. As I mentioned, in places like Rwanda, our team member was the only person in Rwanda who had published on a coronavirus. So he was a huge asset to their team. Similar in Tanzania, taken onto the national task forces. But even earlier in the countries surrounding China, there were, our teams were jumping in and detecting their first cases and consulting on how to get wide scale testing done and contact tracing. And many of those places had much, much better outcomes. In places like Cambodia, also in Thailand, going back to the freezers and finding evolutionary related, some of the closest relatives of SARS-CoV-2 to try and help put that puzzle together about what happened in this terrible crisis that we're living through still. Okay, I would not be me if I didn't try to just give you some information that I hope that you will carry to all of your audiences, that again, it is not the wildlife, it is not the viruses, it is us and our behavior, and we need to protect biodiversity. When we try to wipe out certain species, there are almost always unintended consequences that we sometimes live with and identify right away, and sometimes identify much later that have long-standing consequences. Bats are insectivorous, they're pollinators, they're seed dispersers for endangered trees, because we have fragmented the earth so much that we need large flying creatures to help us keep the planet healthy. But even for many audiences, more importantly, when you try to reduce bat populations, the effect on shedding of viruses is the opposite of what you want. So we've been able to document when bats shed coronaviruses. It's highly associated with when the mothers, the dams, wean their pups. So we really can't encourage people to kill bats, because what happens is they up their reproductive rate. And when they up their reproductive rate, the shedding increases. It also allows for a short-term hole in a very nice habitat in general. And so other bats with potentially other viruses or other variants flood into areas especially when you try to cull in urban areas. So it is absolutely not the right thing to do to cull the bats. We worked very carefully instead with communities and actually came up with a cartoon style picture book that's also has a narration that's delivered at community meetings primarily, and it's been translated in more than 12 languages and delivered in many, many more than that, because the narrator has to read one of those languages but tends to translate it to whatever the local language is where they're delivering it. And this has been incredibly successful. We hear wonderful stories from people saying, thank you. People tell us to be scared, but they don't tell us what to do. And so this really helps them to understand the biodiversity side and not putting themselves at increased risk, but also how to get those creatures out of their house, how to keep them away from their water sources, how to keep their urine and feces out of their food. And that's really what we need to be doing. We need to be learning how to live safely and healthily on the planet, with the planet and all of its inhabitants. So doing this work, as Julia mentioned, has given us the ability to understand what might be possible. And that's what we're calling the Global Virome Project. We did this as a notional idea, but I'm happy to say that the G7 and the G20 and others are starting to consider this as a priority. It's really to get out there and to know about those viral risks ahead of time. Now, we did do the math. And we know that if the whole world started working on this, we could finish this within 10 years. And there are many countries, actually, including China and Thailand, that had started just before the pandemic to do their own work to try and detect all the viruses that they had. But of course, all of that got derailed with the pandemic. But I hope that countries will start to participate at home and also help their partner countries if that help is needed to find these viruses in the future. And if we do that, if we test all the mammal and bird species in the world and discover those viruses, we can inform strategies. And one strategy that we've started that I'd just like to finish with is that we did want to encourage you to participate in a new crowd science platform. We have a new paper out in PNAS last month that is showing the backbone and the underpinnings of the ability to actually start to think about spillover in a different way. And what we did was we looked at all of the risk factors that could be identified for viral spillover for new viruses, and then went out to the experts that work in this realm all over the world, 65 experts, besides the hundreds of people on Predict that work to identify all the risk factors. We had experts that run labs and do public health in 13 countries actually start to say, yes, these risk factors make sense and these are less important. And I'm an expert in this, so you should really believe me, but I'm less strong in ecology or something else, so you might not want to believe my opinion there. We put that all together mathematically and then ranked viruses. We ranked the viruses we found in Predict to that date. We also ranked known zoonotic viruses of wildlife origin as a gut check for whether or not the system was working, and also so that people could make customized watch lists. So we made this tool. You can find it at www.spillover.global, where you can create your own customized watch list, and public health professionals can say, what should I be ready for? Clinicians should say, what should be on my differential list? But maybe more importantly, we can also say these top ones we should be looking at from a perspective of viral detection, viral discovery, viral testing, new vaccines, what research needs to be done, where should we be putting our surveillance dollars, and start to move the needle forward. So just as an example, if I look only at viruses that are in wildlife from viral families that are potential for zoonoses but have not caused zoonoses before, you can see that that customized watch list is dominated by coronaviruses, absolutely dominated by coronaviruses. If I bring you into a different view and I just look at coronaviruses, well, and I let the known zoonotics be in there, you can see SARS-CoV-2 is number one, with SARS, the original, as the next one on that list. And you can start to see how those 31 risk factors I showed you before contributed to the scoring for these viruses. Finally, if you're in Uganda, for example, as we just heard, and you wanted to create a watch list for your own country, you can make a Uganda specific watch list. So you can use all of these filters on the side. And of course, known zoonotics are going to be at the top of that list, like Marburg virus, but then it quickly starts to drive down into other viruses that have been recently found that should be on the watch list that probably aren't yet. So we hope that people would use it. In addition, for a lot of you out there, we want you to participate. And so you can put your new viruses in here. This is how we get to that Global Virome Project reality. People start contributing. And in just a couple of weeks that we have had this live and haven't talked about it very much, we've seen more than 50 people create accounts and start to submit viruses. And you can rank your virus on your own screen and not share it if you feel like you want to do that. We really hope you'll share it, and we will double check everything on the back end. You only have to answer about 10 questions, and then we pull from all sorts of public data sets to populate the information for those 31 factors. And you have the ability to go in and look at what was pulled and correct it if you think the public data set was wrong for your virus and location. So to end, I'd just really like to reiterate some of what you heard from others, but also what I think our team in this One Health framework has been thinking about for moving out of this pandemic situation. And that is that we have to get the political will. We have to be better science communicators. We have to work ahead of time to build trust and relationships to be able to prepare for the future. We can develop and implement a system that works locally, is endorsed and supported nationally, and can be a blueprint for or part of a global strategy. And we heard about some of those global strategies. I'm happy to talk about those in the panel later or in questions. But it means thinking about surveillance and laboratory testing early, as well as being ready to integrate the amazing strength of academics and NGOs as surge capacity for diagnostics. We were months behind here in California, but we had lots of people like you all and like us who could have been doing the PCR. In fact, we were doing it for other countries, but couldn't do it for our own system or our own location or our own communities. So that is crazy, and we can fix that. But I fear, honestly, you can read the rest, but I fear that what I'm finding in these high-level consultations is people are so fatigued now because we've been sheltering so long that there is already this kind of fatigue, apathy that's coming into play. And people are saying, yeah, we should really change that. We should really be more disease agnostic, disease X, whatever. That's too hard. Let's just set up our system that we already have for influenza or this or that a little better. Well, that is fine. We should always do everything as best we can, but that's not going to get us out of the next one. And we really need to maintain this momentum and this ideal to do better going forward. And I do think One Health workforce strengthening is important. We heard about that. We're working, again, USAID has funded the One Health Workforce Next Generation Project, more than 100 universities in Africa and Southeast Asia working together to train in environmental engineering as well as human medicine, as well as veterinary medicine, nursing, pharmacy, really broadening the disciplinary awareness. And those future leaders will help us not get ourselves into this situation again. Thanks.
Thank you so much, Jonna, for another fantastic talk. I greatly enjoyed it, and it's just amazing. The pictures you have every time, the guano farm, I've never seen anything like that. That's great. So I have maybe a bit of a personal question. I mean, you really dedicated a big part of your life to this question, predict zoonotic viruses, pandemic preparedness. And, clearly the science is very strong. You also have a great ability to work with stakeholders and globally in these big consortia projects. So what really frustrated you about the response here? Because I mean, it must have been like, you've been saying this for a decade and preparing for day X, and then day X is coming, and it just seems like the lessons haven't been learned on the next higher level.
Yes, I mean, of course it was frustrating, and thank you for that. Thank you for, but what we know is standing there and stomping our feet, and this comes back from my oil spill days, is not useful. The tragedy is here. Let's move forward. Let's do everything we can to fix it and set up systems to be better in the future. So I've turned my head to try and get into a place of not pointing fingers, not, I have a lot of personal frustrations, but I think that it's proven, working more and more with my communication colleagues and behavior change colleagues, saying I told you so does no good. And it actually makes people stop listening to you. So I feel like, honestly, we as scientists have missed the boat. I teach a course in communication to media and policymakers. I think that's what we need to do. We need our students to be prepared to do better, and we need to work in advance with our media and policy colleagues to really build relationships. It takes a lot of time and effort, and most of us feel like it's not what we're trained to do. But if we don't make our science useful, how do we build the trust with the citizenry? And on top of everything that was going on, I had a very horrible experience through COVID, where people used my head next to the president's head with me saying, from 2016 or 2018, this is coming, look out for coronaviruses. And the president's head saying, no one could have known, we couldn't have known. And it was terrible, because whatever, that stuff happened, but putting my head there made me a target for people that didn't want to hear that. And that was not of my doing. And so then my voice was less heard. People were coming after me. That it was a very enlightening and tragic situation, not just from having worked in this, but seeing human behavior and the weaponizing of health that has led to, so many more deaths than we needed to have, and so much more economic consequence than we needed to have.
Thank you. And if there's one question in the chat, are there any binding or non-binding treaties amongst different countries and governments that are solely focused on zoonotic disease surveillance and detection and data sharing?
Well, non-binding, the Global Health Security Agenda, we were the largest US-funded Global Health Security Agenda project. And despite that, the Global Health Security Agenda is highly focused on pathogen-specific pipelines for testing. So I do think that we need to rethink. WHO had added disease X just fairly recently, before the terrible pandemic, but had not been putting much emphasis on it. The IHR that was talked about earlier, international health regulations, are potentially good step, but I actually disagree with some of what's happening now. We just need to strengthen IHR. Okay, let's make it pathogen agnostic, because IHR was set up for influenza and it's for equity sharing. It's about money. It is not about getting us prepared for or stopping a pandemic. We might be able to change it, or we might need a new structure that's in the same philosophical vein as the IHR, but actually does more for proactive work to prevent and then be ready to work together for pandemics. Whereas IHR, the PIP framework, all of that happened to be talking about how to make sure there's equitable economic benefit. But you saw that really went out the window during a new tragedy, right? So we need to plan for disease X and do it equitably. And we're still not there. And we here are getting vaccinated, thankfully, but a lot of places in our country and most of the other countries around the world don't have access. So we're not even close to out of this yet.
Thank you so much. And last question, if you could change anything, just one thing, to make us better prepared, what do you think is number one?
I think having an integrated system with surge capacity where the academic, government, and NGO research institutes have been pre-evaluated, pre-trained, and are ready to jump into action, so that we have way more capacity than we need everywhere in the world, rather than almost the ability to do nothing but sit on our hands, is completely achievable in a fairly short amount of time, and would probably do the most for us early on.
Thank you so much. Thank you, Jonna. It's been a pleasure having you on.
Thanks for having me.